All right, Chemistry 102. We're going to talk about carbohydrate metabolism in this lecture. And we want to, kind of go through it very carefully and, dissect each one of these sections of chapter 21, as many as we can, and point out, what are the most important parts, for you to understand about carbohydrate metabolism. Because as you can see from this, figure that I took from the internet here, you see that we're going to be talking about some complicated pathways. We've got a lot of arrows, a lot of intermediates here, and there's a lot of enzymes, that we're going to have to take a look at. And I don't expect you to memorize the name of every single enzyme that is brought up in this chapter, but I do expect you to understand what's going on in each reaction. So without further ado, let's get to it. And let's start by talking about ATP, adenosine triphosphate. So chemists love acronyms. We love to have, abbreviations, just like the military, right? So ATP is the name of a molecule, adenosine triphosphate. And we're going to look at the structure of that molecule in a little bit. But ATP is described by biochemists as the cellular energy currency. And ATP plays roles in both, or called what's called catabolism and anabolism. So let's define what those are. It says catabolism is a set of metabolic pathways that break down complex macromolecules, breaks them down into simpler molecules, and in the process, we harvest part of their potential energy for use by the cell. One energy requiring function, so once we harvest that energy in catabolism, what can we use that energy for? Well, one energy requiring function is anabolism or biosynthesis. So what would biosynthesis be? That would be making peptides, making proteins and enzymes, of course, making sugar. If our blood sugar level is low, we might have to make sugar. Right? So these are the kind of things that anabolism is. Okay, biosynthesis. We could also use that energy. Cells use an energy conversion strategy that oxidizes glucose. So we're going to talk about how we would use part of that energy that's harvested in order to oxidize glucose. So break glucose down and harvest even more energy. And what we're going to see is that little bits of energy are going to be released at several points along these, pathways that involve several steps. The energy is harvested. The energy that we get from catabolism is harvested and stored in the bonds of adenosine triphosphate, which we call the universal energy currency or the cellular energy currency. And again, we abbreviate it as ATP. Something that just comes to my mind, while I'm talking to my students is notice you're talking about oxidizing glucose. Well, if I take glucose, which is C6H12O6, and I combine that with oxygen, right? I could have asked you to balance that equation long before we ever looked at chapter 21. Why? Because that's a combustion reaction, right? If you just take glucose and burn it in oxygen, that's a combustion reaction that forms carbon dioxide in water. Now, again, that's not exactly what we're going to do in the human body. We're not going to do a combustion reaction in the same way that we would, let's say with a flame or a Bunsen burner. But you can kind of think of the human body in a way as a big combustion chamber. What happens when we take in fuel in our diet is we metabolize or we break down the foods that we eat. We harvest their energy, but what else do we release? We also release heat, right? Our bodies use that energy, but we also release a lot of heat in the process. Here are some of the types of cellular work that require energy, right? Why would we need energy? And I'm sure that all of you could come up with, a few ideas. I'm going to highlight some of the ones that I think are really interesting with respect to biosynthesis. And I alluded to this during the last slide. Synthesis of glucose, right? How do plants make glucose? Well, they make it during photosynthesis from carbon dioxide and water. That's going to require energy, right? What about when a living organism synthesizes an amino acid or a lipid, let's say? Okay, protein synthesis, that would include enzyme synthesis as well. Energy is also required for what's called active transport. You can kind of guess from the name, active, and there is such thing as passive transport, which does not require energy. The active transport requires energy. Transport of food molecules into the cell. Okay, when we move carbohydrates and proteins into the cell, that requires active transport. That requires energy. What else? Let me look here. It's probably the best one in there anyhow. And then motility, well, of course, to make your muscles move, of course, that's going to require some energy. All right. Anyhow, these are just some, types of cellular work that require energy that I cherry picked. Of course, these are all perfectly legitimate reasons as to why living organisms require energy. Well, let's talk a little bit more about ATP, that molecule, and we're going to look at its structure in a few minutes. But adenosine triphosphate, or ATP, serves as a go-between molecule that couples both exergonic and endergonic reactions together. And if you think about, exothermic and endothermic, I know those are things that you probably studied in Chemistry 101. Those are related to enthalpy. But if you remember exergonic and endergonic, those are related to delta G, Gibbs free energy. Okay, so if you have an exergonic process, it's going to be a process that releases energy. Right, so think about it. Catabolism is breaking molecules down and releasing energy. And then we have endergonic process. Those are anabolic reactions, right? Those require energy being put into the reaction because they don't release energy. And you might be wondering, well, how does, adenosine triphosphate, how does it store energy? Well, in the same way that, a piece of wood stores energy, right? Where is it stored? It's stored in the bonds of the molecules that make up that piece of wood. So ATP captures the energy in a bond, and it's a specific type of bond. It's called a phosphoanhydride bond. So a phosphoanhydride bond. A phosphoanhydride bond is not the same thing as a regular anhydride bond that we saw when we talked about carboxylic acids. But what you might remember is that when we talked about carboxylic acids a long time ago, I don't know why this is APT, this should be ATP. Anyhow, when we talked about carboxylic acids a long time ago, you probably remember acid chlorides. They were super duper reactive. But then after those, we had acid anhydrides, and they were very reactive as well. Now, again, we're not talking about either of those things, but the same word shows up, right? Anhydride, loss of water. So when we make an anhydride, we think of it as being a high energy, having a high energy bond. It's very reactive. So hydrolysis of these high energy phosphoanhydride bonds is going to release energy, and that energy can be used for anabolism. And we're going to see that unfold this evening as we progress through the chapters. So ATP is a nucleotide. Where's my structure? It's over here. So we'll look at it in a second. ATP is what we call a nucleotide. It's a molecule composed of a nitrogenous base. It happens to be purine. So if you've studied, DNA in biology, maybe you've probably seen the molecule purine. It also consists of a 5-carbon sugar, which is ribose, and 1, 2, or 3 phosphoryl groups. If you have 3 phosphoryl groups, this is what we call adenosine triphosphate, ATP. That's the molecule of the hour, so to speak. If you have 2 phosphate groups, that's called adenosine diphosphate. And if you have 1, and this doesn't come up a lot in our textbook, but if you only have 1, that's called AMP, which is adenosine monophosphate. So if we look at the bonds that join these phosphoryl groups to the ribose sugar, to the 5-carbon sugar, the first bond that connects a phosphoryl group to the sugar is actually called a phosphoester bond. Now I'm going to skip ahead here to the next slide, which has the structure of ATP. Here's our nitrogenous base, right? This is our purine right here. It's purine. And then this is our ribose. Maybe get a different color here. So this is our ribose here. Okay, so ribose, so that's our 5-carbon sugar. And here we have a phosphoester bond, and you can see that this bond, if I highlight it in green, this bond is not the same as a phosphoanhydride bond. All right. you can see that the phosphoanhydride bonds are different, okay? And there's two phosphoanhydride bonds, and those are the ones that are squiggles there. Now, when I boxed in, I wasn't boxing in the bond, I was boxing in the functional group, okay? So what I have in the two red boxes, those are phosphoanhydride functional groups. The bond of interest is the squiggle bond, okay? And you'll see that throughout the textbook, whenever she's talking about a high energy bond, she denotes it with this little, squiggle like that instead of just writing a horizontal or vertical line or whatever, okay? And that means that it's a high energy bond. So what does that tell us when we look at this structure? Okay, well, we've got a phosphoester bond, or we've got a phosphoester here. Here's the phosphoester bond. And then we've got two phosphoanhydride bonds right here. Okay, so we've got two high energy bonds in adenosine triphosphate. If we were to break all of this off, so if we were to remove all of this portion here, then, my friend, you would have adenosine diphosphate. You'd only have one high energy phosphoanhydride bond. And if you were to remove all of this, then you'd have adenosine monophosphate. You'd have no high energy phosphoanhydride bond. So that's the structure of the molecule. Again, first bond between, or the first, functional group between the ribose sugar and the phosphates is a phosphoester. So a phosphoester. And then the second and third groups, phosphate groups, are joined by phosphoanhydride bonds, which contain the high energy bonds. So this is just a functional group itself, okay? You should be able to recognize a phosphoanhydride if you see it, sort of, in a question or something of that ilk, or anywhere you see it in the textbook. All right. So there's the structure of ATP. It's a really interesting looking molecule. It's a lot bigger than most of the molecules that we studied in the organic chemistry section of this course. But again, you don't have to have the whole thing memorized. You should be able to recognize it when you see it. I'm not going to ask you to, draw me the structure of ATP. No, I expect you to be able to recognize it, have an idea of what it's made of, and know that it contains two high energy bonds. All right, well, let's move on from there. Structure of ATP. Okay, well, I told you that we can break those phosphoanhydride bonds and we can harvest energy from them, can't we? It says when these phosphoanhydride bonds are broken, large amounts of energy are released. Okay, well, what can we do with that energy? Well, some of it will be released in the form of heat, but some of it can be harvested to do cellular work like we just talked about. They can drive those cellular processes that we just talked about. But we had that whole summary. Okay, here's an example. It can drive the phosphorylation of glucose or fructose. Now, the example that I have shown down here comes, I think, directly from our textbook. It's showing the phosphorylation of glucose. Now, let's think about this really carefully here. Here's beta-D-glucose. We spent a lot of time talking about carbohydrates. We know this is beta because the anomeric hydroxyl is pointing up. Okay, we know that it's glucose. Okay, then we have our ATP molecule. Now, ATP is a big molecule. So in order to, keep things simple, we just say adenosine. When it says the word adenosine, that covers the ribose sugar and the nitrogenous base. Okay? So instead of writing all that out, the person just wrote adenosine in a box, right? Biochemists love to get the maximum amount of work done for the minimum amount of effort, just like everybody else, right? Work smarter, not harder. So there's the ATP molecule. Now, the reason why they put the adenosine in a box is, and simplified it, is because here's the important part, right? This is what we're interested in, are the phosphate groups and the phosphoanhydride bonds. And you can see them highlighted here. I use my green. You can see we have a squiggle bond here, and I've got another one here. Well, when I hydrolyze one of those phosphoanhydride bonds in the presence of water, I end up producing ADP, and I release energy, right? Because this is a high energy bond. What's that energy going to be able to do? It's going to be able to drive the phosphorylation of glucose. Okay, so now instead of just having plain old beta-D-glucose, I've got beta-D-glucose, right? If I count up my carbons, 1, 2, 3, 4, 5, 6. On carbon 6, I have a phosphate. So I get beta-D-glucose 6-phosphate. Now, there's an enzyme that's responsible for this process, okay? And the name of that enzyme is hexokinase. Now, again, you should know that when you see the word kinase, it's going to involve some kind of phosphorylation, right? Adding a phosphate group. And we're going to see this reaction show up again later on in the lecture this evening. All right, so here's, again, a beautiful example of showing how ATP works and how we can harvest that energy. Now, I realize that at this point in the lecture, if you haven't read the chapter or if you haven't watched all my lecture videos, you might be wondering, okay, well, okay, great, you make this molecule and you're left over with ADP. What's the big deal? Well, we're going to look at what the big deal is later on when we discuss the fate of this molecule, beta-D-glucose 6-phosphate, which we abbreviate as G6P. This is going to be an important molecule later on in our lecture. That reaction that we just looked at, okay, where we took the ATP and it looked like it just reacted with the glucose to give you G6P, the glucose 6-phosphate, looked like it just happened in one fell swoop, didn't it? I can promise you that virtually all of the processes that we look at in this class are much more complicated than they appear in our textbook, okay? Some of them are very, very complicated processes. And that reaction, and I'm talking about this reaction right here, okay, you look at this reaction area, you just think, A plus B gives you C and D. Okay, great, just like any other chemical reaction I've looked at. But actually, we can break this down into a two-step process, really. Because first what happens is that the ATP, that high-energy phosphoanhydride bond, gets hydrolyzed by water. What's hydrolysis, right? Hydro, water, lysis is breaking. So water reacts with the ATP, produces ADP, and then it says here P subscript I. I wouldn't expect you to know what that is off the top of your head. But this is what we call an inorganic phosphate. Inorganic. Well, it's inorganic because it doesn't possess carbon, so phosphate. So basically, it's the phosphate group that's going to end up on the glucose molecule, kind of hanging out by itself. Okay, so when you hydrolyze, not only do you get the inorganic phosphate, but you also release energy, right? You release 7 kilocalories per mole. So this is energy released. Okay, now we call this a coupled reaction when the product on one side is a reactant on the other side, or sorry, the product is a reactant in the second step, I guess is the way I should put it. So what happens in the second step, once you've got that inorganic phosphate floating around, it's going to react with the glucose, okay, to form G6P and release water in the process. Okay, great. Well, what else? It's going to cost some energy to put that bond on there, right? Okay, it's going to cost us some energy. So if we sum those two equations, right, if we add these two things up, if that's my equals line right there, what's the same on both sides? The inorganic phosphate is the same on both sides, right? The water is the same on both sides. And if I cancel this out, the 3 kilocalories per mole over here, 7 minus 3 gives me 4. Here's my net equation right here after I've combined them. I get glucose plus ATP, gives me glucose 6-phosphate plus adenosine diphosphate. And hey, look, I've released some energy in the process. So this means that this is an exergonic process, right? My delta G would be negative. And this means it's going to be a spontaneous process. So it's going to proceed to the right. Kind of cool, huh? Now we're kind of looking at, chemical reactions, but in the biochemical world, right? So we can take a lot of the information that we learned in the organic chemistry section of this class, and now we can look at a biochemical reaction, unlike, just in biology class where you might just memorize the names of all these enzymes and all these molecules and intermediates. In this class, we get to really take a look at what these molecules look like along the way. Now, our book, I find, goes into quite a lengthy introduction to this chapter. I'm going to go over section 21.2 kind of quickly because this section is actually covered all of the topics that are mentioned on this slide are kind of covered in chapters 20, 21, 22, and 23. But it says here, overview of catabolic process. In this chapter, we're going to focus on the catabolism of carbohydrates, right? That's going to be our main focus in chapter 21. But the idea is that, as human beings, we don't just consume carbohydrates, we also consume fats and proteins. And so we have to degrade all of these macromolecules, right? We don't eat amino acids in, when we, eat a piece of chicken. It's not individual amino acids. We're talking about protein. So we have to break that protein down into what? We'd have to break that down into amino acids. When we eat a carbohydrate, we're not eating glucose. I mean, we probably do. We probably eat fructose and things like honey and fruits, for example. But many of the carbohydrates that we eat are polysaccharides, like amylose. Right? Amylose is the major component of starch. Amylose and amylopectin. I guess amylopectin's present in even greater amount. But anyhow, what do we have to do with those carbohydrates, right? Those big polysaccharides, we're going to break them down into monosaccharides or simple sugars. And then what about fats? I mean, fats are, triglycerides, for example. They're delicious in ice cream, but they're really big molecules. We have to break those down. We'd have to break those down into fatty acids and glycerol. So now we've got some major players here. We've got sugars, amino acids, fatty acids, and glycerol as well. Well, are all of these, are all of these going to be degraded in the exact same way? The answer is no, and that's pretty clear here. Now, carbohydrates don't provide the most energy in terms of, their quantity. Fats provide the most energy based off of, their mass. They provide double the amount of energy as carbohydrates. But carbohydrates are the most readily used energy source. Okay, in fact, the human brain, uses something like 20 grams of glucose per hour. Okay, so we constantly need to be, feeding our body with glucose, with sugar. So we need to have quick access to sugars at all times. Now, what we're going to see this evening is that what happens to those simple sugars is that they go through the process of glycolysis. That's a really ancient pathway. Okay, you think about the first living organisms could what's the word I'm looking for? The first living organisms had the glycolysis pathway available to them, and what do we see as a product is ATP. So as you can imagine, prehistoric organisms used glycolysis to produce energy. Whereas things like amino acids, they're broken down into molecules like pyruvate. They can be converted into acetyl coenzyme A directly. They can go into the citric acid cycle. If you've studied the citric acid cycle before, great. If you've never heard of it, no problem. Either way. And if you're wondering, like, man, why can you do so many crazy things with amino acids? Well, they have a wide variation in structure, don't they? We looked at 20 different types of amino acids with very different side chains. And so when you study the actual fate and the metabolism of amino acids, it's actually pretty complex. Okay, we look at it briefly in this class, but it can be very, very complicated. And then finally, fatty acids, what happens to those? Well, the glycerol can go directly into glycolysis, but what we're going to see is that the fatty acid portion gets converted into this molecule called acetyl coenzyme A, which can go into the citric acid cycle. And if you're wondering again, what's the citric acid cycle? Well, the citric acid cycle is nothing more than another way to harvest more ATP. So we kind of get two major ways of producing ATP, and those would be glycolysis and the citric acid cycle, which undergoes after the citric acid cycle, we have oxidative phosphorylation occurring, and then ATP. And of course, we're going to talk about all of these things, with the time that we have. Okay, when you eat some food, say you eat a hamburger or something, or a cheeseburger, well, what happens? What's the fate of these macromolecules? How do they get broken down into amino acids and glycerol and fatty acids and then simple sugars? Well, it says here the purpose of stage 1 catabolism is to degrade food molecules into the subunits. So, for example, polysaccharides, like I said, amylose. Polysaccharides like amylose, which are found in starches, they're immediately they begin to degrade immediately as soon as they are in your mouth, right? Because salivary amylase can start breaking down amylose. Whereas we have other enzymes like sucrase, which can break down sucrose, and lactase, which is comprised of what? The lactose and glucose. We looked at that in the carbohydrates chapter. That can lactase acts upon lactose, which is a disaccharide. Maltase is an enzyme that acts upon the disaccharide maltose, which is comprised of glucose and glucose. And then those monosaccharides can be taken up by epithelial cells by active transport. What about amino acids? Proteins are digested, broken down into amino acids by protease. I mean, you think about a protein from a meat, let's say, or from some kind of nut that maybe you ate or something that's got protein in it. Well, as soon as it hits your stomach, it's going to denature it, right? And then it's going to get hydrolyzed by, pepsin and trypsin and chymotrypsin, depending on whether it's in the stomach or in the intestines, right? And we talked about those enzymes in the last chapter. All right. Those are also taken up by active transport. And then finally, the fats are broken down into fatty acids and glycerol. Unlike sugars and proteins, fats are not broken down immediately. Their digestion doesn't begin immediately. Doesn't really start until they get down into the intestines. Okay. First, we break them down into these little globules, right? And that just exposes more surface area. Then they get those globules get dispersed with bile salts, so that breaks them down even further. And it's not until they get acted upon by what's called pancreatic lipase that we start to break them up into fatty acids and glycerol. So it doesn't happen immediately for fats. They don't get metabolized till later on, unlike sugars and proteins. So again, I said salivary glands secrete amylase, or salivary amylase, we call it sometimes, which can digest starches. Stomach acids are going to be able to denature proteins. Pepsin is going to start working on proteins right away. The pancreas is going to secrete serine proteases and other lipases that can act on proteins and fats, respectively. And I talked about active transport and passive transport. So active transport, and again, is for amino acids and sugars, and fatty acids and glycerol move via passive transport. Our book doesn't go into any more detail about that than that. Okay, here are some, a picture is worth 1,000 words, especially to a chemistry student. There's only so many words you can look at before you're like, show me a structure, and there's nothing here that's really complicated, I don't think, for the students of this class. These are all things that we've looked at before, but I just want you to think about it in terms of catabolism now. If we take a disaccharide, let's say it's maltose, for example, we break apart that disaccharide using an enzyme, for example, a glycosidase, right? Glyco, sugar, glycosidase, it's going to be some kind of enzyme that's going to act upon the sugar, so we break that down into monosaccharides. What else? If we have a peptide, now this is just a dipeptide here, but you can imagine, it says here it's a portion of a protein molecule. We can hydrolyze that using a peptidase. We convert that down into amino acids. As I told you, the fate of amino acids really varies by our group. It can get very complicated. And then finally, triglycerides. All right, triglycerides are going to be acted upon by lipases. So we hydrolyze them. Right? We hydrolyze all these esters. We produce glycerol and fatty acids. We can actually get a lot of energy from fats. We can harvest a lot of ATP from triglycerides. Then those small subunits are going to go into various pathways of energy metabolism depending on their structure. It says here that what happens to sugars? Sugars enter glycolysis, that ancient pathway that we're going to talk about tonight, as either glucose or fructose. Glycerol, for example, can also enter glycolysis. Sugars are going to get converted into this molecule called acetyl coenzyme A. They can go directly into the citric acid cycle. Proteins enter at many steps as the carbon skeleton varies, and fatty acids can enter the citric acid cycle after conversion into this molecule called acetyl coenzyme A. And of course, we'll look at the structure of that molecule. Now, you might be wondering about this acetyl coenzyme A molecule. Even if you've never heard of this molecule before, you've certainly heard me talk about acetyl, right? If I asked you to draw me the structure of acetyl chloride, you draw this, right? That's a molecule we looked at many times. how many carbons are there in acetyl chloride? 1, 2. So acetyl coenzyme A or acetyl coenzyme A, however you want to call it, has 2 carbons in it. Okay, and that molecule that's going to have that acetyl group on it. So oftentimes we just write it like this. We draw an acetyl like that, and then we put coenzyme A. Sometimes we make it a little bit more complicated, but what's the point here? Is that we're going to do something with those carbons and with those hydrogens. It says electrons and hydrogen are harvested. The acetyl group is oxidized completely to carbon dioxide, just like a combustion, right? And then the electrons and hydrogen atoms that are harvested are used to make even more ATP during that process that was way at the bottom of that figure we looked at a few slides ago, that process called oxidative phosphorylation. Well, with that in mind, let me find some water, and then we're going to talk about glycolysis. Just give me 1 second. All right, some water. Has anybody here studied glycolysis in biology class? Or looked at it even a little bit, or heard of it a little bit, maybe in high school, or biology at PPCC? I have not. Okay, no problem. No problem whatsoever. Yeah, it's usually, so one student says no, the other student says yes. So just, and if you're too afraid to answer, don't worry, because it's usually a split. It's, yeah, somebody says, yeah, in anatomy and physiology, absolutely, you probably did, yeah. So if you have heard of it, great. If you haven't, that's fine. Somebody says yes in every science class I've ever taken. Yeah, me too. I remember learning about it in 12th grade biology. So it's really important now. So what I want to say to the students who have studied glycolysis in biology, in those classes, they're really obsessed with counting ATPs. I'm sure you've all seen those problems where you have to tally up all the ATPs after the Krebs cycle and everything. Okay, you're obsessed about how many NADHs you get, how many pyruvates, yada yada yada. Now that's important to some degree in this class, but what you're going to see is if you look at the practice problems that I selected from our textbook, you'll see very little of that. Okay, you'll see very little to add it, tallying everything up. In fact, the only time that we look at a big problem that involves tallying things up will be in chapter 23. It'll literally be in our last lecture. I'll go over a problem with you in detail, and it will be abundantly clear how to answer any problem that you're asked in organic chemistry. So what I want you to really pay attention to in chemistry 102 is going to be the structures of the molecules along the way. If there's an enzyme or two that I think you should know, I'll point that out to you. Okay, so glycolysis is also known as the Embden-Meyerhof pathway, rather. Anyhow, so glycolysis, it says here, it's just a way to break down carbohydrates, right? It's a pathway for carbohydrate metabolism. Nothing more than that. Starts with D-glucose. Hey, a molecule we know, right? We know that L amino acids are naturally occurring, but we know that D sugars are naturally occurring. Okay, good enough. It says all organisms can use glucose as an energy source. What else? This is kind of interesting. Requires no oxygen. You think, hey, when you talked about combustion of glucose, well, you said plus oxygen. Well, glycolysis is such an ancient pathway. It was available to living organisms before there was oxygen in the atmosphere. And it even occurred in organisms that didn't have organelles. So they were not as sophisticated as organisms now. They didn't even have organelles. And so glycolysis actually occurs in the cytoplasm, still does. Now, it's a 10-step pathway. I don't expect you to memorize all the steps, but as we follow along, I'll point out what I expect you to know along the way. Okay, it says substrate level phosphorylation gives 4 ATP. Okay. And what substrate level means, it just means that a phosphoryl group is going to be transferred to ADP directly from another intermediate. Okay, and the list of 2 intermediates here, their names aren't important because we're going to look at them soon enough. So we're going to end up with ATPs, okay, and we're also going to make NADH. We're going to look at what that is. It's a coenzyme. I think I mentioned it briefly in the enzymes chapter. And then finally, we're also going to make this little molecule called pyruvate, which is the conjugate base of pyruvic acid, if anybody's interested. And if you're wondering, what's going to happen with all these, well, we'll look at that. Okay, we'll look at that as well. All right. The glycolysis overview. Again, it's an anaerobic oxidation of glucose. It doesn't require any oxygen. Pretty crazy when you think about it. Here's the overall reaction. We have glucose, and notice the stoichiometry. We have only 1 mole of glucose, right? And then it's going to react with 2 molecules of adenosine diphosphate, 2 inorganic phosphates, 2 of this molecule or this coenzyme NAD+. What do we end up with? Here we go. And all my former biology students will be able to tell everybody this, right? You have 2 pyruvates, 2 ATPs, and 2 NADHs. Okay. You also end up with some protons and water. I'm not going to really stress about that a whole lot. But we are going to talk about pyruvate because pyruvate is used in follow-up reactions in order to keep glycolysis going. And then NADH is going to undergo an oxidation process, and we'll look at what we'll take a gander at what that looks like. That reoxidation process has to occur so that glycolysis can continue. All right. Don't get intimidated by looking at this slide. I realize that it's really fine print. It comes directly from our textbook. I'm not putting it in here to scare you or anything and be like, look at all this, okay? Here are the main concepts. We're starting out with glucose, right? The steps go, 1, 2, 3, so on and so forth. Okay, so we're starting out with glucose, and you notice that there's kind of a couple stages here, right? The first stage, you notice that there's just 1 set of reactions, and then something happens right around here, and then all of a sudden you have 2 sets of reactions, okay? And we're going to look at why that is. I can tell you right now that the reason why is because glucose has 6 carbons in it, doesn't it? It's a 6-carbon molecule, whereas pyruvate is a 3-carbon molecule. So 6 divided by 2 would give you 3, and that's why you end up with 2 3-carbon molecules. And again, we'll talk about how that happens, this kind of bridge between the first and the second stage. Other things I want you to notice along the way. You see where the reaction arrows are here? This is a reaction arrow, right? What's the product, of this first reaction? Besides glucose 6-phosphate, it says we make ADP. Okay. Look at the third step. Okay. Besides the product here, and we'll look at what that structure is. What else do you make? You make ADP. Okay, that's good. Well, if you look at the second part, and I'm just going to go over this a little bit quicker, you see what the overall products here are. NADH, I make an NADH here, I make an ATP here, I make another one here, I make another one here, I make another one here, and I make my pyruvate. Of course, there's water as well, right? There's water here, I'm not worried. But those are the products, okay? It told us that we end up with NADH, ATP, and pyruvate. Now you might be wondering about, well, what about these ADPs? Aren't those products? Well, we'll talk about why they don't really get considered as a product. All right, strap your skates on, Gordy. We're going in, and we're going to look at all the steps of glycolysis. Again, we're going to go through them slowly, and we're not going to worry about memorizing every enzyme along the way, except here's your first exception. Okay, it is important that you know the name of the enzyme responsible for phosphorylation in the first step. Okay, in reaction number 1. Meaning, and I pointed this enzyme out to you this evening, hexokinase. We'll put a star by it. You should know the name of hexokinase. And hexokinase is the enzyme that catalyzes the first step of glycolysis. You consume foods that are made up of glucose. You break that glucose down, okay? You break those sugars down. Sorry, you break those sugars down, or those polysaccharides, whatever, to make glucose. So now you've got glucose. What the hell is going to happen to it? Hexokinase is going to phosphorylate it to form what? G6P. Remember that? We looked at this reaction just a few slides ago. This is the first step of glycolysis. I told you it was important. Okay, where does that phosphoryl group come from? Right? Where does this P come from? Well, that came from ATP. That means it's going to cost us ATP in order to do this. You're like, well, hold on. I thought you said we could get energy from glycolysis. I thought you said we could make ATP, and here you are using up ATP. Well, it's like the old expression. You got to, what is it? Scared money don't make money, right? You got to spend a little bit of money to make a lot of money, and that's kind of what happens in glycolysis. It says here, expenditure of ATP early in the pathway works as an energy debt necessary to get the pathway started. But I promise you, there's going to be a payoff in the end. If you go back here, here's this first step. This is all we've covered, okay? We're still not done with the first step. You've made G6P and you produced ADP. So you spent up an ATP. All right, I have another slide here that shows it. I tried my best to color code it. So I think I put all the substrates in yellow, the products in purple, and the enzymes in green. I tried anyway. Here's that reaction. Now here's where it gets good for organic chemistry students, right? You can look at this glucose molecule without any fear. You know what a Haworth projection is. You understand, that there's a difference, in this alcohol at carbon 6, isn't there? This is a primary alcohol, right? It's the most available of all of the alcohols. And so the adenosine triphosphate is going to transfer its phosphate group onto the least hindered hydroxyl to make G6P, glucose 6-phosphate. You produce ADP in the process, and the enzyme that catalyzes the reaction, which again, I want you to memorize, hexokinase. Hexokinase. All right, so that's 1 down, 9 to go. Okay, reaction number 2. The product of reaction 1, which was what? G6P, glucose 6-phosphate, gets rearranged to the structural isomer fructose 6-phosphate by an enzyme called phosphoglucose isomerase. Hey, look, even if I didn't ask you to memorize the name of the enzyme, when we take G6P and we convert it to fructose 6-phosphate, which we call F6P, I'll just kind of scribble it in there. If you look at the molecular formulas of G6P and F6P, they're identical. So of course, that's just an isomerization, right? But now what you're going to see is the product has an exposed carbon 1 that's no longer part of a ring. So you're going to end up with the second primary alcohol. Let me show you. It says here, converts an aldose to a ketose. Look, let me show you what's going on. Both of these reactions that you see in this slide, they're identical, okay? This one is just showing a Haworth projection, and this one is showing a Fischer projection, right? We know that, for example, the aldose form of glucose is in equilibrium with the closed form, the pyranose form. Okay, well, let's look at the reaction on the top. We have G6P. It undergoes an isomerization, and when it isomerizes, you end up with a second primary alcohol over here. Okay, now here is the Fischer projection, and so after it isomerizes, if you look at all of these alcohols here, every last one of them is a secondary alcohol. But look what happens when you convert this aldose, right? This would be aldohexose. Now you convert it to a ketohexose. So this is a ketose. Still has the same number of carbons, right? Oh, it's down here, aldose and ketose. Anyhow, now you've got a primary alcohol. So that's going to be available or open for business, so to speak. And now it's going to be ready for the third reaction, the third step of glycolysis. Let's take a look. It says here, substrate F6P, fructose 6-phosphate. It's going to get phosphorylated again by another kinase. The first one was hexokinase in the first step. The second one was an isomerase. But this kinase is called phosphofructokinase. I think it's kind of, I don't like to say obvious. Nothing's that obvious, but it's kind of obvious, right? Because you're working with fructose. Now you get a bisphosphate. It's called fructose 1,6-bisphosphate. Hey, I mean, the names they speak for themselves, right? Where does that phosphoryl group come from? If you think back a couple slides ago to that 10-step mechanism or that 10-step figure that I showed you, you saw that we used up 2 molecules of ATP in the beginning, don't we? Again, same thing over again. We got to spend money to make money. We're spending ATP early in the pathway, and that's going to kind of get us rocking and rolling. It's kind of getting us to the top of the roller coaster, right? We're going to spend ATP in order to get up here. Once we're up there, we're going to get a big payoff, okay? I don't know why I did that, but anyhow, there we go. All right, let's take a look at the chemistry, the most exciting part. So here's my fructose 6-phosphate. I've got my beautiful primary alcohol all exposed here. My enzyme phosphofructokinase catalyzes the phosphorylation of that primary hydroxyl. And now I get fructose 1,6-bisphosphate, and I get some more ADP. So again, so far all I've done is cost myself energy, right? I've cost myself ATP, and I haven't earned a stinking thing. Well, trust me, it's going to get more interesting. Again, notice every molecule that we've looked at so far, they all have 6 carbons, right? This has got 6 carbons. This has got 6 carbons. So in order for us to get to a 3-carbon pyruvate molecule, and if you've never seen pyruvate, don't worry about it. We'll look at it. But if you've never seen pyruvate, you know, it's 3 carbons. That means we're going to break this thing in half, right? We're going to have to cut that in 2 somehow. And that's why we end up splitting the pathway. It says here, reaction 4. This is one of the most interesting reactions in the whole pathway, in my opinion, because you end up with 2 different products. Okay, it says product of reaction 3, the fructose 1,6-bisphosphate is split into 2 3-carbon intermediates by the enzyme aldolase. And it forms 2 molecules, G3P, which is glyceraldehyde 3-phosphate. You guys know what glyceraldehyde is, right? We looked at the structure of deglyceraldehyde in the carbohydrates chapter. Hey, that G3P or that glyceraldehyde, which is just a phosphate on the 3rd hydroxyl, that happens to be the substrate for the next reaction. But you also end up with another molecule, which is an isomer, and it's called dihydroxyacetone phosphate, or we abbreviate it as DHAP. Let's take a look at what those structures look like. So here's our fructose 1,6-bisphosphate. They just chose to draw it as a Fisher projection. Why? I feel like Mr. Dion, they keep switching between Haworth projections and Fisher projections. Why? It's actually to try to make it easier. Okay? It's because it's easier to see how the molecule is split in half. If we divide it up pink and blue here, you kind of think of cutting that bond. Okay? And we make dihydroxyacetone, and then we have glyceraldehyde 3-phosphate, right? 1, 2, 3, and then we have a phosphate here. Okay, if you look at the Haworth projection, I mean, you can kind of see how if you draw a line down here, you still have 1, 2, 3 carbons here, and here you have 1, 2, 3. But I wouldn't say it's as abundantly clear as to what's going on. So that's why I think, you know, they draw it as a Fisher projection. Anyhow, if you compare the molecular formulas of these 2 molecules, these are isomers. Okay, these are constitutional isomers. So if G3P, or glyceraldehyde 3-phosphate, is the substrate for the next reaction, I'm going to have to take all of this DHAP, right, DHAP, and I'm going to have to convert it to G3P. Reaction 5 is where we take that dihydroxyacetone phosphate and we rearrange it, or we isomerize it into a second G3P molecule by this enzyme. Look, it's another isomerase. They all make sense, I think. You know, an isomerization, same molecular formula, just a different arrangement of atoms. And now we're going to have 2 molecules of G3P. So now you see why the pathway is split in 2, and we're ready for the next reaction. Here's that isomerization. The enzyme, again, this is one of those enzymes I'd never expected to remember. It's called triose phosphate isomerase. If you think about it, this is a carbohydrate, skeleton, this part of the molecule right here, and it would be a triose, right? Because dihydroxyacetone is a carbohydrate. It doesn't have any chiral carbons, but it's still a carbohydrate. What else is I going to say? Yeah, so we're just isomerizing to get G3P. Okay, now we're getting to the second half, right? The second half of glycolysis. Here we go. All right, So now we've got two G3P molecules. You know what? Let me back up. Where was I? I go back to my figure here. I just want to make sure you're on the same page as me. I don't want anybody to be completely lost. Okay. So now we've covered, this part here, and now we've got two molecules of G3P, G3P, G3P. I promise you that both sides of this stuff here, they're identical. Okay, it just happens twice because we divided the molecule in two. Okay, so there isn't like 10 more reactions, only 5 more reactions to look at. Okay, what's going to happen to that G3P molecule, and when are we going to start getting a payout, right? So far, all we've done is lost energy, right, when we used up ATP molecules. The substrate G3P is oxidized to a carboxylic acid by glyceraldehyde 3-phosphate dehydrogenase. Again, an enzyme I would never expect you to remember. But one thing you should know is if we have oxidation occurring, you can't have oxidation without reduction. It doesn't happen. Okay? So when we oxidize the G3P to a carboxylic acid, we're going to reduce NAD plus to NADH. If you don't remember the structures of NAD plus and NADH, we looked at them in chapter 19. Okay? We looked at the structures of both of those coenzymes, and we even looked at exactly where the hydrogen is added. So this H here, that's a hydrogen atom. So what's the definition of reduction? If we're going from NAD plus to NAD with a hydrogen, when we add hydrogens, that's reduction in organic chemistry. Well, we oxidize the G3P, we reduce the NAD plus, and we're going to transfer an inorganic phosphate group to the carboxyl group. This is going to be the first step in glycolysis to harvest energy. Okay? When we make the NADH, we can use that to harvest energy. The product that we end up with is called 1,3-bisphosphoglycerate, and you're going to see that the new phosphate group is attached with a high energy bond, and we can use that to drive reactions forward. A picture's worth 1,000 words. Here's the chemistry that's going on. Now, if you're wondering, you're looking at these structures and saying, I don't see a carboxylic acid here. Well, remember, as I told you, most of these enzyme catalyzed processes are a little more, or maybe a lot more complicated than they're made out to be in our textbook. The G3P is being oxidized to a carboxylic acid, but after it gets oxidized to the carboxylic acid, a phosphoryl group is transferred onto the oxygen of the carboxylic acid. So at some point you did have this, okay, they left it out, and then that carboxyl group got phosphorylated. Notice that that phosphoryl group is attached by a high energy phosphoryl anhydride bond. Look, you can see that we're adding oxygens and we're losing hydrogen, right? This hydrogen that I have circled in green, that's gone, and we gained this oxygen. So it's pretty clear that this process is oxidation. It's pretty clear that when NAD plus, even if we don't have the whole structure of the molecule here, if it's gaining a hydrogen atom, it is undergoing reduction. Okay, so now we've got this high energy intermediate 1,3-bisphosphoglycerate, and that's going to help us drive reactions forward. And we're also going to be able to harvest energy from NADH. Here we go. Reaction 7 is a reaction where we also harvest energy, but it's the first energy harvest in the form of adenosine triphosphate. Here's what happens. We take that 1,3-bisphosphoglycerate with that high energy phosphate group, and it's going to transfer that phosphate group to ADP to make ATP using this enzyme phosphoglycerate kinase. Okay, you've seen kinases enough by now to know that they're involved in the transfer of a phosphate group. The products are going to be ATP and this molecule called 3-phosphoglycerate. Again, this is the first substrate level phosphorylation of glycolysis. Substrate level phosphorylation, again, means when it's transferred from an intermediate 1,3-bisphospho glycerate directly to another intermediate. Here's that reaction. We have the 1,3-bisphosphoglycerate reacting with ADP. We have the kinase, and we're going to transfer one of those phosphoryl groups onto the ADP, and we harvest ATP. Again, I want to point out the substrate level phosphorylation versus phosphorylation. The substrate level means it's going from a substrate, directly to another molecule. When it's not substrate level phosphorylation, that's when you see just like an inorganic phosphate group floating around, right, and being added on. Okay, so that's not substrate level. Anyhow, there we go. Okay, we're getting close to the end now. Reaction 8, we take that phosphoglycerate, it gets isomerized into a different molecule, 2-phosphoglycerate, not much there. We have this mutase, which involves just isomerization, really. So there we go. And it's going to move the phosphate group around from carbon 3 to carbon 2. One of the lesser reactions that I find less interesting. Anyhow, it's just an isomerization of moving a phosphate from here to here, nothing more than that. Okay, well, there must be a good reason for it. The enzyme enolase is then going to catalyze the dehydration, which is the loss of water of our substrate, 2-phosphoglycerate. Sorry, I'm getting tired of talking all day. We end up with this new intermediate phosphoenolpyruvate, which is one of the coolest intermediates in the whole process because it's the highest energy phosphorylated compound in metabolism. Let me show you what it looks like here. The phosphoglycerate ends up losing water. Here are the atoms that are lost. Here's the water over here. And in the process, we make a double bond. So that we get a really high energy, a very high energy intermediate. Higher, what is this right here? High energy intermediate. And you can see our squiggle bond written there. Well, we've only got one step left, and I guess you can probably determine what's going to happen in the last step. The final substrate level phosphorylation. So it's another addition of the transfer of a phosphate group to make more ATP. So phosphoenolpyruvate serves as a donor of its phosphoryl group. It gets transferred to ADP by pyruvate kinase, making ATP. So not only do we produce ATP, we also end up producing a molecule called pyruvate. Okay, the final product of glycolysis. This is a coupled reaction. Remember, I told you a coupled reaction is where you hydrolyze, right, and then you liberate the phosphate, and then it ends up being a starting material for the sort of the second step. So, coupled reaction in which the hydrolysis of the phosphoester bond provides the energy for the formation of the phosphoanhydride bond in ATP. Let's look at the reaction. Last reaction of glycolysis. We take this high energy intermediate here. We do a substrate level phosphorylation. It's catalyzed by pyruvate kinase, and you're left over with a molecule which has 1, 2, 3 carbons. Pyruvate, in chemistry, whenever you see 8, nitrate, sulfate, carbonate, right? Anything with 8 always has a negative charge associated with it. If you have this acid, so CH3, C double bond, clean that up, carbonyl, and then another, carboxyl. This is called pyruvic acid. So pyruvate is just a conjugate base of pyruvic acid. And there you have it, my friends. If we summarize what the heck went on there, I guess we'll get to that in a second. Before I do that, I'll talk briefly, I guess, about fructose, because not only do we produce or break sugars down into glucose, but fructose is, we consume a lot of, or at least most people do, consume a lot of high fructose corn syrup. Of course, fructose is found in fruits. And so fructose is a very important sugar in our diet. So how the heck does fructose get into glycolysis? Well, depending on where the fructose is, it's going to enter glycolysis in different ways. in the muscle. So if the fructose sugar is in the muscle, there's enough hexokinase in there that it's going to phosphorylate it directly to fructose 6-phosphate that it goes, that's an intermediate, F6P, goes right into glycolysis. What else can happen to fructose? Well, in the liver, there's abundant fructokinase, which is another enzyme, and that's going to phosphorylate it, not to give F6P, like I said in the last slide, but it's going to give you this new molecule, fructose 1-phosphate, and then it gets broken apart into DHAP, so dihydroxyacetone phosphate. We know what happens to that. And glyceraldehyde by this enzyme, fructose 1-phosphate aldolase. That's not important. Then the glyceraldehyde, what happens to that? That gets phosphorylated into G3P, and it enters glycolysis. So again, it depends. If it's in the muscle, it's going to get phosphorylated directly, boom, by hexokinase to F6P, and you're off to the races, man. That was one of the intermediates of glycolysis that we looked at already. If it occurs in the liver, there's a different enzyme, fructokinase, converts it into this intermediate F1P, okay? Then it gets broken apart into DHAP and glyceraldehyde. Then the glyceraldehyde gets phosphorylated to G3P, and you've already got the DHAP, right? And boom, you're off to the races, okay? So it just depends on where the fructose is. But you can see how fructose can be, even depending where it is, it can end up in glycolysis. This is just a cartoon showing, depending on where it is. Okay, so let's take a look. Picture's worth 1,000 words. We have our fructose molecule right here. It's being acted upon in the muscle by hexokinase. It makes F6P, fructose 6-phosphate. Hey, again, that was an intermediate that we saw in glycolysis. So wow, it just goes right into glycolysis. Okay, if it's in the liver, then this different enzyme, fructokinase, is going to act upon it to make F1P. That's a new intermediate. We didn't see that this evening. But then it gets acted upon by this aldolase, breaks it apart into, hey, DHAP. Everybody knows what that is, right? And then it gets converted into glyceraldehyde, which undergoes phosphorylation to give you G3P. And walla walla bing bang, it ends up in glycolysis. Just like that, right? So that's what can happen with fructose. The regulation of glycolysis, I'm going to go over this quickly with you. Look, I'll level with you. What you need to understand for Chemistry 102 is really simple, okay? This slide makes it look super duper complicated. If we think about the glycolysis pathway, if we concentrate on 3 steps, okay, there are 3 steps involved in regulation of glycolysis. For example, if we don't need energy, say the living organism doesn't need ATP, why the hell is it going to cause glycolysis to occur? That costs energy, right? Why would it bother? Okay, so in order for a living organism to regulate glycolysis, it can be regulated by 3 enzymes at 3 steps, steps 1, 3, and 10. What was the enzyme that was used in step 1? Well, it was hexokinase. In step 3, it was phosphofructokinase. And the last one was pyruvate kinase. What would make glycolysis, progress at a faster rate? Why would a body want glycolysis to occur? Well, in other words, what's going to activate glycolysis? The answer is this, and all you need to know is this. The pathway is going to be highly activated when there's a deficiency of ATP, right? Okay, if the body is ATP deficient, come on, it's going to push glycolysis forward to make it more ATP. Now, if you're like, well, if there's no ATP, what's left over? ADP. I agree. Our book barely mentions adenosine monophosphate. I mentioned it at the beginning, but adenosine monophosphate, or AMP, is the precursor to ADP. Look, all you need to know is that adenosine monophosphate doesn't have any high energy phospholate hydride bonds. So look, if you have a high quantity of adenosine monophosphate in your organism, what's that going to tell the organism? What kind of signal is that going to send? It's going to say, hey, there's no ATP. Get this thing going, Charlie, and move it. I like to move it, right? Okay. So that would be an activator. Again, that's all you need to know. You don't have to worry about the intermediates here. If you want to, try to memorize every intermediate along the way and explain, that's totally fine, no problem. But again, I think for Chemistry 102, as long as you know that AMP is going to drive the reaction forward, that's good. Well, conversely, what's going to stop glycolysis? When is a living organism going to say, whoa, 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 there's enough, Well, again, we look at steps 1, 3, and 10. The bottom line is this. If you already have enough ATP, why would glycolysis even happen, right? What's the point? You've got enough energy. Why do you need to start making more? Okay, so that's going to be an inhibitor. Other inhibitors would be the intermediates produced in each of these steps, like G6P that's produced in the first step. If the body has a lot of G6P, it's going to send a message and say, hey, I've got enough energy, or I've got enough G6P to make ATP. There's no reason why I need glycolysis to occur at the moment, right? If I have the intermediates citrate, or if I have too much acetyl coenzyme, same thing happens. But for me, in Chemistry 102, at the ACS level, what you need to know is what's in the yellow and green circles. That's it. Okay, and all of these enzymes, remember we talked about positive allosterism and negative allosterism? Well, all of these enzymes, hexokinase, phosphofructokinase, pyruvate kinase, right? These are complex molecules, aren't they? They're all allosterically regulated. So what does that tell me? That tells me that all of these intermediates, like adenosine monophosphate, these are going to bind allosterically to any one of those three, or phosphofructokinase or pyruvate kinase, and they're going to activate it, right? They're going to open up the binding site and just say, come on, let's go, make more, okay? Whereas ATP is going to bind allosterically, but it's going to close the active site, right? Kind of cool when you think about it. It really ties together chapters 19 and 21.